So first things first, we're going to go through how to do the hang drop slide technique. So first, what you want to do is you want to obtain a cover slip for each of the specimens that you want to visualize. So here we go. We have three cover slips. Now I'm going to do an extra cover slip here because there's a reason for it, and I'll tell you in a second. So we have the three. So it's really important that you label everything. Remember, right? So here we have Euglena, which is 1, Amoeba 2, Paramecium 3. Each of the pipetters are labeled with a number. See, 1, 2, 3, and they're in their own containers so that we can keep them from cross-contaminating each other and whatnot. So on the hang drop slides, the concave slides, I'm going to label these 1, 2, 3. So everything corresponds to one another, right? So now what we're going to do is we're going to take the immersion oil. So this is immersion oil, the same oil that we used for the microscope to visualize our organisms at 100x. We can use it for the hang drop technique too. Now the way I want you to do this is I want you to prime the tip, right? So you're priming the tip. You can see that there's a little oil now on the tip. So what you're going to do is you're just going to touch. You're going to dab a little bit of oil on each of the 4 corners. You see, and so you can dip back into, like, if you get too much in one area, you can dip back into it and you can spread it around. So here I have all 4 corners for the Euglena completed. So now the amoeba will do the same thing. So we're going to prime the tip. You don't want a drop to come out. You just want enough to be able to move it around the various parts of your, various corners of your cover slip. Now we're not putting anything on this cover slip, like immersion oil wise. So that completes the amoeba. Then now for this one, we're going to prime the tip again. Didn't have anything there. So now once it's primed, then you can move those droplets of oil around. Okay, perfect. Okay, so now let's talk about the concave versus convex, right? So concave, think of it like a cave, right? You walk into a cave. So concave would be like this, concave, where the slide is like this, and you're going into the slide basically, like a cave, right? Convex is when it bubbles out. So for our purpose today, we're using a concave slide. So you want to make sure that the concavity is facing up. So sometimes what you can do is you just run your finger, kind of over it, but it's facing up here. So we can actually walk into the slide, so to speak, because it's the concavity is facing is pointing up. Okay, so we have our 3, and they're all oriented in that manner. So now what we need to do is we need to take our samples. So the first step of the exercise today is to take a drop of each of these organisms. So let's take I'm going to take the lid off of the Euglena, and I'm going to press the dropper, go in, and take up just a little bit, not too much. That's quite a bit actually. But so now what we want to do is we want to put a small drop in the middle of the slide. You see how, or the cover slip. So you see how there's a small drop, and then we're going to just set the dropper off to the side. Now that drop may be a little too big, but we'll see what happens when we put the slide over it. So let's do that right now. So let me move this over. Let me put this over here and show you that. So show you that. My gloves got stuck to it. Okay, so we have that. So let's move it up just a little so you can see it. Okay, so this is one. So we're going to take now the concave slide, and we're going to face the concavity down so that it forms like a dome over our droplet. So now I'm placing it over, and I don't want to drop the slide down, right? So I just want to put it over it and then flip it. It makes contact, and then you can see the drop, and it's sitting in the middle of the slide, okay? So now let's take a look at what happens when we look at it under the scope. So let's put it on the scope. So I'm going to show you that. So here we have it on the scope. I'm going to position it over the light source. And I'm going to, and we're only going to use 4x. We don't have to, we can go up to 10 if we want or whatever, but we don't have to go to 100. We don't have to go to 100 with this because this is a pretty large organism. So we're increasing, we're raising the stage. And right now, as you notice, I have the condenser all the way up, and you can see a lot of background noise. You see that? So when I drop the condenser down, it clears it up, see? So now you can see a bunch of euglena. They're all there. So now I'm switching to 10. The software, again, is auto-correcting, right, for everything. And now I'm fine focusing. And as you can see the euglena moving around, and you notice how it moves. It's not moving in a straight line. It's kind of zigzagging around, and that has to do with the type of mode of locomotion, right? So it possesses a flagellum. Let me lower the light a little bit, and let's see. And again, it's auto-correcting, so, but we wouldn't be able to see the flagellum necessarily because you would need to stain it to see it well. But refer to the sheet that I've posted in Blackboard so that you can see that. So that's the euglena. Okay, so once you're finished viewing the organism, you're going to then take the hang drop slide, you're going to swish it around in disinfectant. So here we have a container of disinfectant right here. So you're going to take the slide off of the stage. You're going to slip off the cover slip. You're going to swish around the slide, and then you're going to wash the slide, after you've done this, wash the slide in soap and water and reuse it. That's what you would normally do in lab. But for today, I have a separate slide for that. So I'm just going to set this to the side right now on this paper towel. And we're going to go back to our next organism. So we're going to look at the amoeba. So let's go back. So here we have the amoeba. So now let's talk about the reason for this cover slip. So the reason why I wanted to have a second cover slip for this particular organism is because this organism, excuse me, this organism settles to the bottom of the container. So you don't want to disrupt or agitate it too much, because if you do that, you'll end up not getting anything when you actually draw up the sample. So I'm going down to the bottom. So I'm pushing the dropper, right? I'm pressing it, and I'm going down to the bottom of the container, and I'm going to slowly try and suck up some of the debris that's down there without disturbing too much of the bottom, like without mixing it. Okay, so now I'm going to take this and I'm just going to put it on here. So once you have a large drop on this cover slip, then you don't necessarily need to go back to this container anymore. So we'll cap that container. So now we're going to pull straight from here to then place onto here. So let's do that. So I press my bulbous pipette just a little bit. I go down to where I see some debris, and then I slowly, carefully place that on my cover slip. This organism is very hard to get, so sometimes I really try and get some of that debris, because the organism usually, the amoeba is usually in that debris. So I want to make sure I have some. So I think this drop is a little too big. Right. You just kind of got to play around with it a little. You get it. Okay, so we have a drop there. Whether or not it has an amoeba in it, that's a different story. So here we have the drop. So now we're using slide 2, the concave slide 2. We're going to place this over the drop, touch all 4, flip it carefully. So now you can see the drop is hanging. It's there in the center. So now I'm going to put this on the stage, drop the slide down. Now I'm going to show you as I raise the stage. So I'm going to raise the stage right now. Raising it up. Okay, so you can see that there's a bunch of stuff on here. Well, right now, none of it is the amoeba. There's a lot of other protozoan they put in there to feed the amoeba. So we need to kind of go through it and see if there's anything actually amoeba related here. So let's check this. Let's check this area. No, I don't see it there. Don't see it there. Keep looking. And it's hit and miss, like I said. Sometimes you get it on your first try, other times you don't, and sometimes you have to repeat. You have to repeat it a few times. So right now I'm not seeing it. Yeah, I'm not seeing it in this one. So let us go back to, well, let me get another hang drop slide, and let's try this one more time. So bear with me here. Entertain yourself with the protozoan that you're seeing on the screen. Okay, so let us go back to doing this again. So here we have our concavity is here. Set this to the side. So again, this is 2. And we're going to get our cover slip, place it, put our 4 drops, prime it. 1, 2, 3, 4. Okay, there. So we got our 4 drops. Then now we're going to take our slide, or no, sorry, our dropper, and we're going to go back into this drop that I did. I'm just going to dump it all there. That's a big drop, right? That's too much. So put it there and then suck off. Hopefully we don't get rid of the debris because I want some of that debris. Okay, so we got that. Now we're going to take this, we're going to flip it over, we're going to touch it, and okay, so this is what happens when your drop is too big. So the droplet actually makes contact with the concavity, the bottom part of the concavity, and it shoots the sample off to the side. I mean, we can still look at this, but it's not going to be as it's not going to work as well as a true hang drop would, because it's now spread out, and so it may dry out a little faster and whatnot. So, but so let's now look at this. If I don't find it on this one, we'll move on to the next one. But here we have some more of that debris that I was talking about, and yeah, I'm not really seeing it here. There's no amoeba here on this one either. Let's look a little more. Yeah, I'm not finding it. Let me drop down. Oh no, I'm already down to 4. So this is 4x right now. So I don't see it. So let's for the time be. Oh wait, no, is that? Let me zoom in on that. So now I'm on 10. Let's see, maybe it's here, maybe it's not. I don't think so. It doesn't look like it is. And sometimes the company will actually ship out to me a sample that has none in it. So it's really hit and miss when it comes to the amoeba. But okay, so let's move on. So we'll put this behind us for now, just for now. And let's do the paramecium, the last organism. So we have our concave slide already labeled. Now we're going to take the dropper, we're going to press it just a little bit, and we're going to go into the sample, take some of it, and then place that in the middle of our slide. Okay, there. Okay, so we have a drop. So now we're going to take our hang drop slide, flip it over. Do not put it down on the table. So you want to just touch it, and you see how the drop is nice, and you can see it. It's perfectly formed, and it hasn't made contact. You flip it carefully. You see the hang drop. So now we're going to take the other slide off the scope, and we're going to place the paramecium now on here. So let's move down to 10 or to 4. Sorry. Oh, and see, so we have a paramecium here. Let me see if I can focus it a little better for you guys. And they're quick. They're really fast. So there's one in here. Let me see if I can find it again. Oh, there he is. Okay, so he's really booking it, right? He's moving quickly. He's all around the field of view here. I'm trying to focus in real time. So remember, this is a liquid, right? This is a thick liquid because remember, it's a drop. So he's got multiple layers that he can swim through. And because of his mode of locomotion, he's able to traverse that droplet, and so he goes in and out of focus. Oh, I think I just saw him. Yep, there he is. He goes in and out of focus. So they're very quick. You can see the oral groove on it. It's very cool. So check out the sheet that I've posted about this, so you can look at the different types of internal structures it possesses and the means of locomotion for this particular protozoan. So let's try and jump back to the amoeba. So what I'm going to do is I'm going to do something real quick. It'll help me hopefully get an amoeba for you. So I'm going to do something. So let's see if it works. I'm going to take a centrifuge tube, if I can get one. Here we go. I'm going to take a centrifuge tube, and I'm going to put the amoeba inside the centrifuge tube. Okay, so here's the centrifuge tube. I'm going to try and help myself by getting an amoeba this way. Let's see if it works. Okay, so I have some of that. Let's take some more of this. The pressing going in, being careful not to disturb anything while I'm taking from the bottom of the container. Okay, putting it inside. Perfect. And I'm capping it. So now I'm putting this in a centrifuge quickly. I don't want to waste too much of your time. So right now I'm over the centrifuge, and I have the tube, and I place it in the centrifuge and just spin it down for a few seconds. Once I've done that, I'm going to take my bulbous pipette and I'm going to press it before going into the centrifuge tube. Okay, so you're going to see me do that right now. So I open the microcentrifuge tube, and I get rid of anything that's in the tube, the pipette, the bulbous pipette. But first, let me set up my slide. So I'm going to put the 4 drops of oil on the corners, and I'm going back to each drop if the drop is too big, and I'm just pulling it to another corner. I have it set up, so now I'm going to open my centrifuge tube, and I'm going to push the pipetter, go to the bottom of the centrifuge tube, and then try and suck up some of that debris that's at the bottom, and then put that debris in the center of the cover slip. So you can see I'm trying to get as small of a drop as possible there, but it's kind of hard to do that with a bulbous pipette. So I put a larger drop, and you can see that it's a bit of a mission. So I'll work with it for another few seconds here, and I think I'm just going to go with this drop. Now, just be aware that this won't be a true hang drop, because the droplet's going to immediately touch the concavity, as you can see right there, and it sends it to the side. But I just want to see if I have an amoeba from the debris that I was able to centrifuge out. And now we're looking at it under the scope, and you can see that there's a lot of other protozoan that are swimming around, and that's mostly just food for the for the amoeba that they place in the solution. So I'm looking around to see if I can find an amoeba somewhere on the slide. And I'm looking. I can see it a little clearer than you can here through the eyepieces. So don't worry if you can't see anything right now. I just found one, so now I've gone to 10x, I believe, and here you can see the amoeba, and you can see the actual cytoplasm inside the amoeba. So just be aware that the amoeba is not doesn't have a defined shape, right? So it uses a false foot. It creates a false foot by streaming its cytoplasm. You can see the cytoplasm moving and extending this pseudopod to the top left portion of the screen. And the amoeba is very light sensitive, so it's trying to escape the bright light of the scope. So it's moving away from the light. Another cool thing about the amoeba is the way that it consumes other protozoan and whatnot. So it'll actually surround the protozoan. We've seen this before in lab, actually, where it's surrounded the protozoan with its pseudopods, and it has taken in the protozoan and encapsulated it, and then basically broke down the protozoan within the capsule that it's created, the vacuole that's created within itself, and then just consumed the protozoan. So, but the amoeba's it's a really neat looking protozoan, and it definitely has a means of motility. I don't know if you I know that you can see it. It's definitely moving from point A to point B, and it's doing so very slowly, but it's doing it. So let's just watch it for another few seconds. And definitely look at your information sheet that I've posted in Blackboard regarding this protozoan, so that you can see the various parts with that it possesses. Okay, so now we're going to go to the next portion of the exercise, which is looking at live organisms. So I have a stain bin set up, and I have 2 slides. Now, these 2 slides, I'm going to put a drop of DI water on them, and we're going to use this DI water in order to emulsify the bacterium in the droplet. So first things first, though, let's label our slides. So I'm labeling one E. coli and one M. luteus. So okay, so now that both slides are labeled, the slides are labeled, they're on the stain bin. We're going to put the drop of oil. So this is just, a drop of water, the DI water, sorry. And again, this is the wet mount. So the wet mount's different from the hang drop technique, in that the wet mount does not hang the water droplet. Instead, a cover slip presses down on it. So it's different. So anyway, I'm flaming my loop, and I'm getting ready to place the first organism. So we're gonna look at first the E. coli, and we're gonna take it from this plate here. So I open the plate, I touch the loop to the area where there's nothing to cool it, and then I grab a little of the organism and I place the plate down. And now I'm going back to the droplet of water that I placed on the slide, and I'm emulsifying the organism in the droplet. So I'll tap it a little to try and get some of that E. coli off of it, off of the loop, so that it's in the solution. And then next I'm going to take my loop and I'm going to flame it again. Remember, aseptic procedure. Burn off anything that's on the loop. And once I've done that, I'm going to set the loop to the side, and now let's turn our attention back to the slide. So I just grabbed a cover slip. I'm now going to take the cover slip and I'm going to position it on a 45-degree angle with the actual droplet that we placed the organism in, and I'm going to simply just drop the cover slip. And by doing this, it pushes the air out from beneath the cover slip, allowing you to not see air bubbles and whatnot on when you look through the scope. So now we're going to put it on the scope, and we're going to take a look and see exactly what E. coli looks like. So here we are at 4x magnification. I'm right now going, with the stage. I'm raising the stage up to bring it into focus, and you'll see that I can see like debris and stuff on the slide. At this stage, you can't really see the organism because it's much smaller than most of that larger debris that you can see here. So we'll see it a little better once I switch to the next power. So it's really important when looking at organism that's not stained to make sure that you manipulate the light on the scope. You don't want too much light. You want to be able to darken the field of view by dropping your condenser down, or lessening the amount of light going through the diaphragm, or something, or even the dimmer. You can turn the dimmer, but you can see here at 10x, you can see the dots, little dots in the background, and they're kind of, there's some movement going on there. Whether or not it's true or if it's just Brownian movement, that's a different story. You'll have to look at that at a higher magnification. So now I'm switching to 40x, which is the high dry, remember, objective. And with the 40, we're again going to have to adjust the light, because right now it's just way too much light. And you can see that I'm fine focusing through it, and there's so many layers to this because remember it's a droplet under a cover slip, so there's a layer of fluid, and the organism is throughout that layer of fluid. Even though that layer is very thin, there's movement. There's different levels to it. So as you find focus, you'll see through different levels. So now I'm switching to 100x magnification, which is the or objective, and this will allow us to see it a little, much closer. So I'm switching to 100, and I'm adjusting the lights and whatnot, and I'm fine focusing, and you can see that there's something there at this point, and I've put I've already put a drop of oil on it, so I'm now fine focusing through the sample, and you can see what you see there. Does anyone recognize the shape of this organism at all? You may not be able to see at this stage. I may need to adjust the light a bit further. So let me see what I can do about that. So let's drop the light down a bit, and let's find focus. And with unstained bacteria, it's really difficult because, like I said, they're not stained with anything and they're transparent. So the light, you really have to mess with the light and get the contrast to show the actual organism. You'll see, though, when we start the staining lab that it'll be much easier for you to see these organisms. But this is live, we're not staining, and then yeah, so take a look at this and see if you can determine if it's true or if it's Brownian movement. And remember what true movement is and what Brownian movement is. And if you can, note the shape of the organism. So take a good look at it and see if anything is apparent to you there. So the movement, just take a look at the movement. Is it from point A to point B, or is it just kind of a to-fro Brownian type movement? And again, this is just your presumptive evaluation of the motility of this organism. Okay, so now we're gonna now switch gears, and we're going to go back to our slide preparation that we did. We're going to flame our loop again, make sure that is sterile before we actually go into our culture tube. So the organism now that we're working with, I'm pulling from a stab, since I don't have a slant available. I'm pulling from a stab M. luteus with the loop. So I touch the surface of the stab first to make sure that the loop is cool. And then I'll grab the organism, but I'll flame the tube, put it to the side, and now head over to the slide. So on the slide, I have my droplet of water ready. So I'm just gonna emulsify the M. luteus, and you can see some of the organism came off in that droplet. You can see a little cloudiness there. My head's not in the way. And then we're gonna take a cover slip, same way we did with the E. coli. And we'll take that cover slip, and we'll again angle it at that 45-degree angle, and just quickly just drop it, like release it, and it'll force all that air out from beneath the cover slip. So let's now take it to the scope, and let's load it in and try and course adjust, right? So we're at 4. The only objectives you should use course adjustment, and bring the stage towards you. And as you do that, you will see something that mostly looks like debris, because, like I said, any organism is going to be much smaller than what you can first see when looking under the 4x objective. So now I'm looking at it, and I'm kind of trying to course, using the course adjustment, trying to focus using that, and I'm kind of fine tuning it with the fine focus until I get it to a point where I think that I'm seeing it. So now I switch to 10, and here it's much larger, and now you can see that they're very tiny dots, and there's a ton of them, right? Because I put a bit of the organism on the slide. I'm trying to now look to see if maybe there's a little more separation, so that we could possibly see it a little better. But But for the most part, I put a lot of organism on this in this droplet. So most likely the concentration of the organism is pretty consistent throughout because it's underneath the cover slip. So now we're going to move to the 40x objective, and with the 40x objective, again, remember these are unstained organisms. You need to make sure that you manipulate the light to be able to visualize the organism. So 40x, we're doing that. We're fine focusing. Remember, because we're at 40, so we're fine focusing, and then now we're switching. We're switching to the 100x objective. So I've already placed the oil on it, and now we're fine focusing. But I have to adjust the light, of course, and we're moving around the cover slip to try and see if we can get the bacterium into focus here. So you may need to play around with the fine focus. Make sure that the oil makes full contact. Increase, decrease the light. Sometimes the mission to really find these unstained bacteria. So you just keep working with it. And here I'm fine focusing. I'm moving the slide around a bit, trying to get something going here. Now you can see that there's stuff there, right? There's a movement to it. So try and determine if it's Brownian or if it's actual true motility. Is it point A to point B? Is it all going in one direction, which would then indicate current flow, underneath the cover slip? So take a look at that. But I'm dropping the light down, and wow, as you can see here with even less light, you can really see the shape, and in even some of the arrangements. So the actual morphology of the organism is visible, but it's not as visible as it could be if you were staining it, with a stain, which we'll do, like I said, in a future exercise. And so take a look at this, see the shape, see the movement, and determine from your presumptive observation on whether or not this organism is or is not motile. And just be aware, whenever you're doing a wet mount, that you're working with live organism. So you have to treat this as biohazard. You don't want to get any of the liquid from, what you're working with on anything that, your hands or anything. And if you do get it on your gloves, you'll want to change your gloves before you touch your scope and stuff like that, because you don't want to contaminate your lab manual, your partner, I don't know, like the materials in the lab, the scope, whatever. So just be aware of that. So now I'm going back to the Bunsen burner, because we're doing the last part of this exercise, which is the stab inoculation. So this stab that we're doing is a motility stab, and motility stabs, as you're aware, have less agar than traditional stabs. They have 0.2% instead of 2% agar. So I'm uncapping the tube, and I've already taken the organism, and now I'm simply going to go into the tube, and then I'm going to quickly push down to the center, not going completely to the bottom of the tube, but almost to the bottom. then capping the tube, placing it aside, and flaming my loop again, right? So it's always important to do flame before, flame after, remember. And also note that I flame the actual opening of the tube before I went into it, and then after I went into it, I did the same thing. And I'm doing it here again for the other organism. So the motility stab, the purpose of the motility stab is to either confirm or reject your initial presumptive observation of the motility. This will tell you for sure if the organism is motile or not, and we'll talk about that in the next part of the next exercise. We'll discuss the results from today. So so we'll flame our stab again, and then we'll set that to the side, and pretty much that's it. So that's exercise 3, and yeah, we'll see our results next lab. Thank you.